Thanks for joining us for another episode of Biotech and Life Sciences CEOs. Recently, we've been talking about the speed at which change is occurring in the life sciences. And nowhere is that more clear, I think, than in the field of mRNA. By now, I think everyone knows or must know that the COVID vaccines were created literally days after the genome of the virus had been sequenced. But that fact alone is so overwhelming that I don't think we've still really processed the meaning. Our guest today is Pooja Tiwari. She's the founder of Arnav Biotech, dedicated to the design and manufacture of both custom and cataloged mRNA for use in therapeutic, vaccine, diagnostic, and even agricultural applications. Pooja received her BS and MS degrees in biochemistry at Nagpur University in India and her PhD from Alabama State in 2014. As a postdoc at Emory University, Pooja's research focused on flu and HIV viruses. For more than 10 years, she served as a contributing reviewer at the International Journal of Nanomedicine. Pooja has published at least 28 peer-reviewed articles on subjects including HIV and RSV, and has received at least 2 patents for her work. Pooja, let's start by, can you please tell us how you came to your early interest in science as a youth in India, and how you came to US as a student at Alabama State?
Yeah, hi Tim. Thank you so much for inviting me over for this podcast. I am just going to be honest, I'm a little nervous, but at the same time, this is really exciting opportunity. So I'll just start with a little bit of story about me. So I'm Dr. Pooja Tiwari. I did my BS and Masters from Nagpur University in India, and then my PhD in Alabama State University. So it's going to sound a little cliche, but like a lot of kids my age watched what's one of the most important movies in, I think, recombinant DNA technology, Jurassic Park. And and at that time, I, of course, didn't speak as fluent English. So this movie was dubbed in Hindi. And I watched the very important scene of the movie where this amber, which has a mosquito in it. And I saw that movie and I was like, oh my God, this is amazing. You can take a mosquito's DNA. And I did not know what a DNA is at that time. And you can make dinosaurs out of that. And I went home just literally jumping. I don't know why I was jumping because half of the things I didn't understand. But my sister was studying BS microbiology. So I went home to her and I was like, you know what? Like, you can really take this DNA and we could make dinosaurs. And I thought that I could make dinosaurs. So that is the earliest memory that I can relate to, like what got me excited. But of course, as a young kid, I was always injecting my doll with like all sorts of things, and I played doctor with my very healthy doll. But anyways, fast forward a few years, I tried my luck to be a doctor, but apparently it's too competitive in India. So I instead, pivoted towards doing my bachelor's in biochemistry and biotechnology. And we had a lot of these cool experiments where I actually visibly saw a DNA when we extracted it from, I guess, like spinach leaves or whatever, and then, of course, bacterial cells. And we were doing just regular lab experiments where we are cutting DNA and then like trying to see what happened. So this was really cool for me that I got my memories back from Jurassic Park. So then it got my interest more and more, um, basically like cemented that this is something I wanted to do. And then I explored different options. And somehow through one of my professors, whose daughter was in Montgomery, Alabama at the time, I got introduced to my PhD mentor, Dr. Sri Ram Singh, and they had this amazing PhD in microbiology program. And that's how I landed up in Atlanta and then Montgomery, Alabama for the next 5 years of my PhD. And I am more than fortunate to have had a mentor like Dr. Singh, who is also our CEO for Arnav Biotech now. And that's how my journey started. And I cannot be more thankful and fortunate to be here and talking to you today.
Great. I know that I'll have my grandson watch Jurassic Park as soon as possible.
You
How,
should.
how did you initially become interested in mRNA?
So after my PhD, of course, as is with anybody, in academia, my plan was to go into postdoc, do a couple of years of postdoc, and then probably try my luck in getting a faculty position somewhere. So I was looking for my postdoc opportunities, and I came to Emory University under my PI, Dr. Francois Bollinger, and they had a grant that was together with Georgia Tech. So when he was moving out of Emory, I switched to Georgia Tech for my second year of my postdoc, and in that lab we were doing um, lots of mRNA, and that was like really interesting because I had always been like a DNA kind of person, and then I was like, well, mRNA is really cool, and we started doing a lot of work on mRNA based antivirals, mRNA based therapeutics. So the main project that I was kind of heading was expressing anti-RSV antibodies, palivizumab, which is still the prophylactic antibody given to the kids who are severely infected with RSV. So we were trying to express this instead of using directly palivizumab. We were trying to give mRNA intratracheally and then see how much expression we can get. The whole idea behind that was that from the time palivizumab given intramuscularly reaches the lung, it's decreased by around 2000-fold till it's in the lung. And we were trying to counter that by directly delivering into the lung. And we saw really good expression of palivizumab and also, protected against RSV infection. So that's was really the moment where I thought of like really getting involved more and more into mRNA. And more I worked on it, more I fell in love with mRNA. And contrary to what like there's been views about mRNA, I have seen very first and very positive things about mRNA, and I guess that's how the journey started.
Okay. So what happened as far as that work with RSV is concerned?
So that work was actually published in a journal called as Nature Communications,
Right.
and that actually kind of led to multiple different grants expressing other antibodies. For example, there's a bunch of broadly neutralizing antibodies against influenza and also against HIV that we expressed, and there were like subsequent publications that we kind of worked and then found very good protection against HIV and then also influenza. And some of that work continued to go on after I had left my postdoc position. But then on the side, we also developed some other therapeutics with mRNA. But that was one of the time that I guess like the grant that we had from DARPA didn't get funded or extended for continuing that work. And then COVID happened. So we had another project which was using a CRISPR protein called as Cas13 to be used as a therapeutic against influenza and SARS-CoV-2. So I guess like for good, we kind of got more involved in that work, and that's also a patent pending and continuing in the research lab. And I had pivoted out of the lab by the time, and I continued to do other things with RSV.
Okay. And did that have anything, any input, say in Moderna's recent work in RSV, which I guess they were only nominally successful with?
Right. So Moderna's work is actually trying to like express, antibodies, not antibodies, but like it's a more of a vaccine. RSV is kind of, it has its own challenges. I don't want to go into the details of its, virology for the sake of keeping the conversation simple. But there has been a lot of progress, definitely. And even though the vaccine may not have been where we would have expected it to be, we still could learn a lot from that. And of course, there are like other vaccines and other therapeutics that Moderna and other companies are continuing with mRNA. And I'm very hopeful that something positive will come out very soon.
Okay. One of the other things that we've been talking about lately are the two principal impediments, I think, to progress in the life sciences at this point, and that is manufacturing and delivery. And from the mid 70s, scientists had some understanding of mRNA and a sense of its potential usefulness, but it really wasn't until the late 1980s, I think, that scientists were able to manufacture it successfully. Are there particular things that you can point to in terms of its manufacture that have improved over those early efforts?
Yes, of course. So obviously, like you're right that so first of all, I just want to like reiterate the fact that mRNA is not something that doesn't exist in our bodies. Just on an average day, every single day, our bodies are making thousands of different types of mRNAs because that's how we make the information that's in our DNA into the proteins that we need for functionality. So mRNA is quite natural. It exists everywhere in all living cells. But the fact that we are able to make this and make it do the job we want to do is attributed to a lot of factors. So first of all, making an mRNA is one thing, but having it to express the protein we want at a stable scale inside the body has a lot of factors. For example, discovery of not discovery, but ability to use lot of viral proteins. For example, T7, it's a bacteriophage, or SP6. These are the bacteriophages whose enzymes are used now in vitro to make the mRNA at scale. And there are companies who have actually invested a lot of money, infrastructure, and resources in making these enzymes work better with higher efficiency. And then on top of that, the naturally occurring base that an mRNA has is uracil, which is somehow has a lower stability inside a living cell. So what scientists have also come up with is another modified nucleoside called as the N-methyl pseudouridine, which is found in one of the archaea, which is like the third domain of life. And they have tried to incorporate that into the mRNA to make it more stable. So there is definitely a lot of improvements in terms of like this, the synthesis of the mRNA, and then the way it's delivered, and then also the way it is capped and tailed. So all of these factors taken together have made the mRNA to be what it is in a Moderna vaccine or a Pfizer vaccine. So all of these have attributed to the development in related fields of biotechnology. For example, even the DNA template synthesis has become so much inexpensive compared to what it has been like maybe 20 years ago. So all of these factors together have led to the improvement of synthesis of mRNA as well as its delivery.
Okay, so help me here for a second, because my understanding, at least initially, that the mRNA, which is produced for science, has a very, very slight difference from the mRNA which is in our body. Is that true? And what difference is it? I guess it's something that makes it work, is my understanding, but I certainly could be wrong.
Sure. So essentially, as far as the design of the basic design of the mRNA is concerned, it's almost the same as we have in our body. The only difference that we are seeing now is the way it is capped. So basically, I don't have a drawing, unfortunately. I should probably have had these, but the design of an or a simple structure of an mRNA has different components. It has like the untranslated regions where different enzymes bind. So the mRNA can be made from DNA into an mRNA. And then once the mRNA is made, it has to be stabilized by using enzymes which are called as capping enzyme, which add like a 5-methyl guanosine cap on the 5' end, and then also there's poly A tails on the 3' end. So what happens is that when naturally occurring, these processes happen with the help of enzymes that exist in our body. But when we are trying to make this in vitro or in tubes or in an industrial setup, we add these enzymes separately. So the slight differences that you see in the human body and the ones that are made in the lab are how this cap is added. So for example, there is something called as process wherein the cap is added while the mRNA is being transcribed. So that's one difference. But again, essentially, it does not alter the quality or the basic structure of the mRNA whatsoever. The only difference in terms of the Moderna or Pfizer vaccine is the use of modified nucleoside or a modified uracil compared to that of a normal uracil. But it does not, I just want to emphasize this fact because I want your listeners to get this fact very, very right, that it does not whatsoever make this mRNA stay in your body forever. Even though it is stable, it does not mean that this mRNA is going to stay till we are alive. It's going to get degraded after maybe like a couple of days. And then once it makes the protein, is the protein that gives us the immunity, not the mRNA. And even though it's using the modified nucleoside, it is still something that exists in nature, and it's not completely artificial. It's just that it does not have in human body, and we are using it from another live organism. But essentially, the mRNA is the same, or if not identical, it's similar to that of the mRNAs which we produce in our body every single day.
Right. And I think it's important. I can't believe it's important, but it is important to note, because I hear from random people sometimes that, oh my gosh, it changes your DNA. No, it does not. I'm not a scientist, but even I know that.
So I'll take one moment, and I want to emphasize that it's just not the common folks. And I think I feel bad when somebody like from a science fraternity or somebody from an mRNA field kind of tries to be like patronizes these people who have very valid reasons, right? It's, as scientists, our job to understand their fears or like whatever their concerns are. They're not invalid. I totally understand. And unfortunately, I have had this question from a scientist who was just not agreeing that, like, oh, how can you make sure that the mRNA is not going to get integrated into the genome and mutate? So anybody who's asking this question is valid. I understand that. But as an mRNA scientist, I want to make sure and emphasize the mRNA, even if it is a replicating or a non-replicating synthetic mRNA, does not have the basic components that are needed for it to integrate into the genome. So there are multiple factors. I want to highlight a couple of them. First of all, for any piece of nucleic acid to integrate into human genome, it will need a couple of different proteins. For example, I'll quote the example of HIV. So why does HIV integrate into our human genome? Is because it has a couple of enzymes, for example, integrase, and there are like enzymes which will cut the viral DNA at a specific position, also cutting the human genome at a specific position and making it go inside the human genome and integrate. But both of these two things are DNA, right? And the mRNA we are talking about is ribonucleic acid, which has a sugar, which has a ribose group, and the DNA does not have that ribose sugar. So those are another differences. Second thing is the DNA has A, T, G, and C. These are the 4 nucleotides that the DNA has, whereas the RNA has AUGC, or A, N1-methyl pseudouridine, and then guanidine and cytosine. So all of these are not compatible to bind in a way that it will integrate into the human genome and make it mutated. So these concerns are valid, but at the same time, scientifically, it is not possible for the mRNA to just integrate directly as is. It will have to first convert into a DNA, then it may very remotely have at least a chance to get integrated, but that's still not going to be possible because it does not have the essential enzymes, essential components that are needed for it to get integrated into the host genome or human genome.
Yeah, right. Can't get into the nucleus of the cell. So you're manufacturing what I understand are functional, reporter, and then what you call decorator RNA. I like
Yes.
the decorator term.
So it's one of the hidden members of our team. They have designed this term. So essentially, what we call is reporter RNA is any RNA, any mRNA that one wants to use for reporting the delivery of mRNA. So, for example, a green fluorescent protein or luciferase protein. So these are the proteins that we manufacture, which anybody can use if they want to test their delivery vehicles and see where in the cell it goes. So those are some of our products. The reporter genes are, of course, we have some functional reporters, for example, beta-galactosidase. That's an enzyme which people use quite a few times for testing expression of a clone gene in a bacterial cell. So blue versus white colonies kind of thing. So we have that. We also have CRE, Cre is actually one of the mRNAs that we offer to our customers who are using Cre-Log's mechanism of recombination, wherein they can test whether a cell has gotten a particular mRNA or not. And Decorator is actually our newest baby. I think we have a lot of data, a lot of good data from that. And I think like in the next week or two, we will be putting out an official release of this product. So as an mRNA scientist, we have always used luciferase or nanoluciferase as a mRNA to test our delivery vehicles. Unfortunately, in some of the cases, this is not an ideal mRNA because detecting this requires a luciferin assay, and then you also need to have a plate reader with a luminometry capability. So that kind of like makes it difficult to be assessed, and also you need to detect it in a certain time frame. So what we decided is to give scientists more liberty. So they are not restricted by using only fluorescence or luminescence to detect their mRNA's delivery, but instead we offer 5 different tags. So it has like different components where people can use any one of these 5 antibodies to stain their cells or tissues, and then detect this at their time, their convenience. They can detect this as and when they want. So it has HA, CMYC, and like a couple of other tags. So people are free to choose whichever method they want to detect their mRNA.
Is that a proprietary product?
Yes.
Must be, right? Okay.
Yes.
And what differentiates not only each of the functional reporter and decorator, but in particular, what differentiates the decorator product from what is currently available in the market? Why is it special?
So I think the USP of this product is the freedom to choose between which method one wants to use for detecting this RNA. So I think the freedom to operate in deep tissues versus cells or live cell imaging, all of that is possible with this one product. So instead of switching from a fluorescent protein to a luminescence or to a colorimetric method, people can choose whichever antibody they want to stain and can identify. Moreover, it has like also like a VHH antibody, which is also our proprietary, like a tag. So people can use any of these VHH antibodies as well to detect this protein. So I guess like the unique point, USP of this product is the freedom that it offers to our customers.
Okay, and then can it be further customized?
Yes.
And what sort of requirements might there be that might imply a customization?
So I think like so most of our customers that we have seen reach out to us for this particular product are the companies or the research groups who are exploring their own delivery vehicles, because currently when people are trying to develop their lipid nanoparticles or polymer based nanoparticle delivery, they always rely only on luciferase, and then sometimes the luciferase signal may vary depending on the deep tissue that they are trying to assess it. And then also, at least in my experience, the luciferase antibodies have not been so great. So the detection after fixing these tissues may become difficult with these luciferase antibodies. So that's what we are offering, that we are offering people to choose what antibody works best for them. And we have a range of these tags available. So they don't have to rely on one tag. They can detect it in multiple different ways. So I think that separates it apart from the currently available market products.
I see. Okay. And is there a difference in design as to whether the RNA is going to be delivered by an AAV or a lipid nanoparticle or whatever it happens to be? Do you have to design them differently?
So again, that's something the researcher has to choose from. But most of our customers don't deliver mRNA using AAV. So far, they have all been using lipid nanoparticles. But again, people can customize these depending on what they are trying to do. But essentially, at least on the mRNA side, we don't have to modify it depending on the delivery vehicle. It is, I guess, it can be used with different types of delivery vehicles.
Okay. And I guess AAV is has, since COVID, become a lot less popular. Safety issues, all that sort of thing. Is that correct?
Yes, true, unfortunately. So I may be biased, but I do have a little of my own personal opinion about AAB. I'm not going to share that to avoid any statements that don't want to defend. But at the same time, AAV has its own restriction. Definitely, like the side effects in terms of like toxicity. And I just read about this report about one of this young kid, unfortunately, who had a very good I forgot if what genetic disease it was, but he's been like doing very well with the treatment itself, but then had unfortunately um, I didn't read the whole report, so I don't want to like use my words, but had some side effects or maybe even passed away because of the cytotoxicity. And that's exactly what my concern is with using viral vectors. But again, I don't want to like say this explicitly because there is a lot of other applications where viral vectors have been successful. Um, so yes, it does have its own issues. And that probably is why mRNA has really proved as an alternative. Maybe, one has to like carefully look into like what works best for what application.
Okay.
But that's one thing that is associated with AAV, and so far we have not seen with mRNA.
Of course, quality control must be very important. My guess is that you work with the FDA and other regulators to try and maintain that quality control. What can you tell us about special efforts at Arnav in terms of quality?
Yes. So one thing I would really emphasize is that because our company is established by people who have firsthand worked from a very small scale of mRNA to up to a gram scale of mRNA, we have firsthand experience to scale this up, and our motto here at Arnav is to make mRNA accessible to each and every single person on this earth, not just like limited to one part or the other. And for that, we are trying to make it more accessible, more better in terms of quality, better accessible, and then also stable. So we do put a lot of effort and a lot of emphasis on the quality of our mRNA, and all our mRNAs are quality tested, not just for its purity, but also for its functionality. So every single mRNA that leaves our door has at some point been tested for all of these components in-house. So most of our competitors might just give you a simple agarose gel electrophoresis as a quality control, but we don't do that. We go above and beyond. We not only like test it for all different make sure there is no impurities, make sure that it's working fine, make sure it is expressing in multiple cell lines. All of that data is always associated with any single mRNA that leaves our door. And we also like emphasize in making this more as less troublesome for our customers as possible.
Okay, and cost. I mean, the field of RNA manufacturing must be driven by cost, design, and speed. And you talk about access, so clearly cost will be, particularly outside the Western world, so important in terms of access.
Yeah, definitely. So what we are trying to, of course, like people always associate things that are inexpensive to low quality. And that is exactly what we are here to change. Our job here is to make the most high quality mRNA that is out there. And I don't want to name names, but our mRNAs have actually proved to be better than some of these competitors that are way bigger companies than ours is. But our mRNA beats their mRNA in terms of quality and stability. As far as the cost is concerned, because we are a very small team, we're a very localized team, we were able to maintain a profitable, but at the same time, cost that is affordable for most of our researchers, because the researchers that we work with are small teams themselves. And we also work with small biotech companies who are trying to venture into mRNA therapeutics, but are not just willing or ready to put the full investment into that. So we are here to change that. We are here to change this notion that something that is not too expensive will not be good. And all of these companies were doing fine, but since COVID happened, I have realized that they have jacked up their prices. And also the delivery timing has almost doubled or tripled in some cases.
Really?
Yes.
Wow. Um, so, there are a number of now CROs in the RNA manufacturing field. Aside from cost, and you allude to quality, are there other things that might distinguish one from another?
Yes, of course. So one thing we really take pride is that I guess again it is attributed to the fact that we are a local small business. We take every single customer, every single client as a personal collaborator, not just like a customer. So we emphasize on collaborative spirit in the approaches, because a lot of these customers not only come to us for our off the shelf products, but most of these customers come to us with the help they need for designing their mRNA. So there is still like a lot of things that are like safeguarded amongst the mRNA community. A lot of people don't want to share what makes their mRNA better, but we are here again trying to be very unusual and a company that takes pride in sharing the knowledge that we gain through our process and through learning through our process development. So we take pride in being one of the probably the only company who not only provides just synthesized mRNA for the customers, but actually help and guide them navigate the field of mRNA therapeutics so that they leave Arnav with a product that is optimized for success. Of course, like in life sciences, nothing can be guaranteed. I mean, we have seen like a lot of things that look very good in phase 1, but will not turn out good in phase 2 or whatever.
Yeah.
But we take pride in making sure that our customers are at least well equipped when they leave Arnav with an mRNA in their hand. It is mostly optimized to at least safeguard that the product will be reaching where it possibly can.
So I know, Pooja, you're a modest person, but maybe you can share with us one or two success stories for Arnav. Perhaps where a client came to you with an especially vexing problem, and you were able to solve it for them.
Yes. So unfortunately, because they're a VC backed company who have a lot of their internal companies that they're still like trying to figure out a way how to come out. So I cannot name them, but these are one of our biggest clients, and we're very thankful because that brought a lot of revenue for us. But when they came out, they simply had this protein-based vaccine, which they thought is working good in their hands. And this was right when the pandemic was at the peak, like around 2020, early 2021. And at that time, they didn't want to like invest fully into mRNA-based vaccine, and they really just wanted to test the waters and see how the mRNA version of their vaccine would look. And of course, these were one of our clients who did not have any background knowledge on mRNA-based vaccines, or like how even an mRNA will be designed. And I think they were even smaller team at that time as well. So what we were able to do is we actually came up with like 8 different iterations of their on their the vaccine, and could help them navigate that, like what are the optimal variations of their protein vaccine would be and how best they would look like. So there was a lot of skepticism at that time at their end, that like because we were such a small team, we were such a young company, and I think we were still building our website at that time. So it was just like a mutual, I guess, leap of faith. And I'm so grateful to them for like trusting us, and we were able to design their vaccines. And we very recently, in the last couple of weeks, got the data from their collaborators in somewhere on this earth, where they actually found that our mRNA vaccines are doing as good, and in some cases even better than their protein vaccine. So we are keeping fingers crossed and just waiting for the time when we will be able to share this more openly. And we are actually they not only gave us this one project to work on, but we have a MSA with them, a multi-service agreement. So we will actually be their, hopefully, longer term partner to collaborate on multiple future vaccines.
That's great. So, do you ever um, take sort of a piece of the equity of a product from a client in exchange for your work rather than just a wholesale payout? Or do you ever do that sort of thing, particularly
So
since
we are
you're
very
a
open.
CMO?
Yeah, so we are actually very open. So we want to establish ourselves as the mRNA therapeutics company who, with a good heart, I don't know if it's a very cliched line, but we are a company who's, as I mentioned earlier, right? Our goal is to make mRNA more democratized, and not just like hold on to this knowledge. And in India, there is a saying that like knowledge expands with sharing. So we are definitely into this business to make sure it's expands beyond boundaries of countries, boundaries of continents. So we are open, whatever works for our clients. We definitely have to find a mutual ground that works for both of us. But this last example that I quoted, we have kept the contract very open. So of course, and they are, I guess, like somehow it's just like a match made in heaven. So they're like us. So they are also very kind in putting us in their IP and whatnot. And when the time comes, we'll be re-evaluating this situation. But we're not really claiming equity in their company or anything, but we'll probably come to an agreement which is probably beneficial for both of us. But we are open. Whatever works for anybody, we are open to discuss that.
Do you think it might be possible someday to use AI to automate the design process?
Absolutely. Although that is one thing that makes me nervous because I used to be the least computer savvy person. But that, of course, had to change. But I'm not an expert on AI, but looking at all the things that people are at least putting out on the social media that the AI is able to do, definitely AI is going to improve the mRNA design. A lot of companies are already doing this. There are like a lot of companies who have already started using AI and machine learning for designing, at least discovering and designing therapeutic molecules. So I'm sure big companies, especially, I'm not surprised if Moderna is already doing it. I'm pretty sure they are already doing it, and it is here to stay. So I think like we should maximize the AI capabilities and come up with better designs of mRNA and better therapeutics.
So I wonder, in preparation, I read somewhere that the Department of Defense was trying to create a process to miniaturize
Yes.
RNA manufacturing process. Why would they do that?
So
What,
it's,
you know,
sorry, go ahead.
what do they hope to gain? And is that something that broadly the industry is looking to
Yeah,
mean?
so I'll answer this question because this actually was one of the things that, again, like being a very small team, we did not apply for that. But I think DARPA, if I'm correct, either DARPA or one of these Department of Defense agencies had put out a grant application where they were trying to have something called as nucleic acids on demand, NOD. So essentially, first of all, are they doing it? Yes. Second, like, why should they be doing it? It's again the same thing, access to an mRNA, right? Just imagine deep down, because always there will be a case where will be some human being stuck in some remote part of the world, be it military or be it some other maybe healthcare workers, somewhere somebody might be in a position where they are um, prone to different kinds of biological threats. So in that case, it might be not feasible to manufacture a vaccine on demand and by deliver them in time. So to change that, if we had just imagined like a printer, right? So I can't imagine a time back in the day where you had to like go to like a specific computer, take your jump stick and then like print something. But now we can do whatever we want to print just sitting where we are sitting and like just hit a command and like the things come out of your printer. Now just imagine this situation for a vaccine. Why one shouldn't do this, right? Like imagine if you had all you would need. Imagine the situation where we already had a COVID vaccine protocol, right? And let's say there is a SARS-CoV-3, God forbid, we already know how to design an mRNA vaccine for SARS-CoV, right? All we will need is a sequence of this mRNA and something that can make the template, and then the mRNA and encapsulate it into a lipid nanoparticles. So if all of this thing is miniaturized, we would we can easily skip the supply chain, or we can skip the delivery part, right? Every corner, or every, if not every healthcare center, at least major centers would have one of these vaccine printers, if you may call it, at their disposable. And all they would have to get the communication from somewhere, central command, is the sequence of the antigen or sequence of the therapeutic molecule that they need. So yes, I think it's there's a lot of scope. There are some people that I already know are working on this technology. So one of the bottlenecks for mRNA based vaccines was synthesis of templates, right? So this client that I was talking to you about, we actually were still under the time that we promised, but still there was a phase where we just kept on waiting and waiting because we didn't have the DNA synthesis capacity. So all of these bottlenecks, the delivery chain, and then like the cold chain storage, all of these issues will be taken care of if there is way to make these vaccines at the point of care. So yes, I think like that's a good idea for anybody to be investing on.
Wow, that's really interesting, particularly when I interviewed a fellow that is working to, I'll use the word miniaturize, a sequencing machine so that it can sit on a shelf in every hospital. It seems to be, particularly with the prospects for personalized vaccines, especially because you need speed, right? You have to get it to the patient. And if it's right there, why you have a better chance of doing that. Maybe we could talk for a minute about those, just kind of get some input from you. I know it's not something that you're involved in right now directly, but personalized cancer vaccines, they require customization and speed. Do you have a vision for how the Arnav platform could better fulfill those requirements?
So definitely, because we are again, I keep saying this unfortunately, but we are a small team and we are limited by resources, and definitely that's one hold up why we are not currently investing into the cancer vaccines per se. But of course, mRNA based cancer vaccine, I think it's not a distant reality. I think it's going to happen pretty soon. Again, the debate shouldn't be around whether it can be done. It would just be around like how effective that would be. And cancer just is a whole another a complicated disease that needs multiple different aspects to be effective. And the Moderna's vaccine, along with that PD-1 therapy, I think looks pretty promising. So I would not be surprised, and maybe within the next 5 years, I hope so, that there will be some kind of significant breakthrough in the cancer vaccines. As far as Arnav is concerned, yes, we are thinking about it. We're not investing actively in that field yet, but we do have collaborators that reached out to us who, of course, are not an mRNA companies, but who are oncology-based companies, and they are interested in trying the mRNA versions of their therapies. So we have actually one of these startups in a stealth mode at Emory that have, we started talking about it, but of course, when somebody already has a, candidate in pipeline, they like to take it for farther first, then to seek a different version of that modality. But that's something we are actively looking into. So we don't want to do this single-handedly because that would need a different kind of setup and expertise. But we are open to anybody who is interested in using our therapeutic pipelines, our platform for testing cancer vaccines or cancer therapeutics. We are definitely open for collaboration, but we are also actively initiating these collaborations on our end as well.
And what about, do you think that it could benefit an area like CAR T?
Absolutely. I definitely think that mRNA is definitely going to. So one thing I really want to like iterate as an mRNA scientist myself, that mRNA may not be the thing that replaces every existing thing, but it will definitely be the thing that improves or like enables these therapeutics to work better. So definitely CAR T is definitely one thing.
And safer. I mean,
yes.
could mRNA theoretically improve the safety outlook of,
I believe so. I definitely believe so. So for example, earlier we were talking about the AAV based delivery vehicle, right? So with viral vectors, sometimes repeat dosing becomes a problem
right?
because of their additional toxicity issues or their like immunity that comes for the virus itself. Like, so those are the cases where I think having an mRNA would definitely come up, would offer definitely better, you know, upper hand to these technologies, really.
Right. Well, thank you so much, Pooja, for your time. I've learned so much today. And it's a field that, we're hearing about every day and we keep learning about every day. So it's really been a pleasure to get to know you and to learn more about mRNA with you.
Thank you so much, Tim. I really appreciate it. And thank you so much for having me over.
Thanks for tuning in to the Life Sciences and Biotech podcast. We'll see you in the next episode. The information contained in this website and podcast are purely informational and not considered investment recommendations. Tim Dougherty's participation in Biotech Insights is separate and apart from his role as an investment advisor representative. Nothing contained herein can be construed as a recommendation or endorsement of any of the companies discussed. Tim Dougherty also has no financial affiliation with any of the companies mentioned in this communication. Tim Dougherty makes no representation that the information contained in this material is accurate and is under no obligation to update this information as changes occur.